Go ahead.
Okay, good afternoon. Welcome everybody to the ARK Diagnostics webinar. Afternoon for some folks. I know still morning on the West Coast, and if you're joining us from Europe or other parts of the world, good late evening. So thank you for joining us today. This is one of our first webinars at ARK Diagnostics, and we hope to do several more in the coming months and years ahead. So we'll get started. We have two very good speakers today, so you should be hearing about advancing methotrexate testing, clinical utility, and analytical performance improvement with ARK Methotrexate 2. So my name is Pankaj Sinha. I'm the VP of Sales at ARK Diagnostics. I've been here for a short amount of time and really enjoying my stint here. So I will take on the lead in introducing the speakers and also be happy to take your Q&A questions with my colleague Nico, who's helping me. Both of us at the end of the presentations will take your questions. So you should be able to see a Q&A box on your screen. So please feel free to put in your questions as the speakers speak, and we'll try to get to as many questions as we can, keeping the time in mind. Before we dive into the main presentation, I wanted to give you a quick snapshot of who we are and what we do. Many of you might know us well, and some of you not so well. We are a 20-year-old California-based company based in the beautiful Bay Area, and we are a leader in TDM and toxicology therapeutic drug monitoring. Our diagnostics is now present in over 60 countries through our partners and distributors. We have a proven track record of developing clinically impactful diagnostic tests. That's something you'll find with ARK Diagnostics is we don't necessarily make me-too products, but really high-quality tests for reference labs and hospitals. I think our biggest area of expertise and our core competency lies in cancer drug monitoring and anti-epileptic drug testing, which you will hear about today from the two speakers about the cancer drug monitoring. Most hospitals and reference labs highly trust us for the quality of our test to provide accurate patient monitoring. Our portfolio is very robust in toxicology assays with several patented IP as well, like gabapentin. We are expanding that portfolio, and you will see a lot of good, solid new tests coming out during the course of 2026. What is also very interesting about ARK Diagnostics that we have a big presence in veterinary diagnostics through our global partnerships as well. This is probably a good segue to give you a sense of where we are headed in terms of a growth strategy and the IVD landscape. Last year, Archimed, which is a private equity company, they purchased ARK Diagnostics, a majority stake in ARK Diagnostics. And I do want to mention that Archimed also has Stago Diagnostics, which is really a major brand and a company for coagulation, hemostasis, and several others like Aspirion Therapeutics, which does lipid tests as well. So just to give you a sense of our expansion and growth, we will be growing quite a bit, and you'll hopefully hear from us quite a lot. We will be bringing in new tests and new product lines to address unmet clinical needs, and we intend to scale more beyond what we already do in our global commercial footprint. We'll be investing in new next generation technologies, and you will find us coming to you and sharing our expertise in specialized diagnostics as well. So short and sweet, that was my introduction about the company, and I want to get to the meat of what we intend to do today. And really, we want to provide a platform for leaders in the laboratory medicine space to share their scientific data and share the knowledge throughout the world. And hopefully you find benefit in that. These scientists are leaders in what they do, and hence we wanted to request Joe to share his scientific data and inputs. And if I may introduce him before we get started, he's a nationally recognized leader in clinical chemistry and lab medicine, and he's serving as a professor of lab medicine at Yale School of Medicine, and is also director of the lab there. He's board certified by the American Board of Clinical Chemistry and is a fellow of the ADLM, American Association of Diagnostic Lab Medicine Academy as well. Joe started his education as doing his Bachelor of Science degree from the American University in Beirut, followed by his PhD in Cleveland State University in Ohio, and he completed his postdoc fellowship at the Cleveland Clinic in 2014, where he further developed his expertise in clinical chemistry and lab diagnostics. He is highly internationally recognized researcher and educator, and his work focuses on improving lab medicine and patient care through innovation. His research interests include the development of indicators for monitoring clinical lab performance, investigation of biomarkers, including acute kidney injury. In addition to his academic and clinical leadership roles, Joe serves on the editorial boards of clinical chemistry, clinical biochemistry, and is also currently the chair of the International Federation of Clinical Chemistry and Lab Medicine, which is IFCC. And he serves the chair of the Faculty of Advisory Council to the Dean of the Yale School of Medicine, beg your pardon. He's a prolific contributor to the field and has authored more than 150 peer-reviewed publications and was honored with the 2023 Young Investigator Award by the IFCC. I think that's around the time I met Joe. I've known him for some time now. Interesting fun fact, he is originally from Lebanon, and Joe is known amongst his family and friends for making exceptional homemade grape leaves, or at least that's according to his wife and his oldest daughter. For those of you who didn't know, it can take about 5 hours to actually make good grape leaves, or at least worthy of Joe's signature, chef abilities. So with that, I will pass the baton to Joe and share his presentation. Thanks, Joe, for making time for us.
Thank you, Pankaj, for the lovely introduction. Hi, everyone. Thank you for joining us today. And I'll start by sharing my screen so we can begin the presentation. So what I really wanted to focus on today is discussing briefly the clinical utility of methotrexate because it's relevant to the analytical performance characteristics we're seeking with the assay. First, I'll say I have the conflict of interest to disclose is that I'm receiving an honorarium from ARK Diagnostics for the stock. Otherwise, I have no relevant financial conflicts of interest. First, just a brief introduction. Yale, most of you think of the old buildings we have. Obviously, this is Yale University on the left side, the School of Medicine building, which is affiliated with the Yale New Haven Hospital. On the right side is the Department of Lab Medicine's building, where basically in Yale New Haven Hospital's chemistry lab is housed, where I work with our staff. And we are a full-service laboratory that offers automated chemistry testing, so where we have the examples of methotrexate loaded and running on our systems, as well as specialized mass spectrometry-based testing, which I'll also mention during this talk. So to begin, then, what is methotrexate? So I thought that would be a good brief introduction, just so about the history of this drug. Essentially, it was first reported and synthesized in 1947. It's a small molecule of about 450 grams per mole, and it was originally known as emethopterin, which you can look up, and you might find in old information in the literature refer to it as such. And it was further developed in 1949 after scientists realized that targeting folic acid may have some benefits in treating cancer. And so in the 50s, it was further developed and used to primarily treat acute lymphoblastic leukemia. In the 60s, dermatologists realized there's also benefit for psoriasis. So then it became used in that setting in low doses to treat psoriasis. In the 80s, then clinical trials demonstrated benefit for rheumatoid arthritis. So today, essentially, there's broad clinical utility, and the main FDA-approved uses for it are in autoimmune diseases, as we discussed, with the benefits that are known for arthritis and psoriasis, as well as in oncology, with benefits demonstrated for leukemia, lymphomas, and solid tumors. So how does it work, and why is it used in these settings? So from the perspective of benefiting cancer, its anti-proliferative effects really operate by inhibiting folate conversion to tetrahydrofolate, because it inhibits essentially the action of dihydrofolate reductase. So through this inhibition, tetrahydrofolate cannot then go on and contribute to the development of essential compounds for DNA and RNA synthesis. And Importantly, there are other known pathways which methotrexate acts, not only to be anti-proliferative, but also it has anti-inflammatory properties and other, and they're still discovering more and more mechanisms. So I'm not going to go through the rest of this because it's not a biochemistry talk focusing on the ways methotrexate work, but there are multiple ways that it acts, not just on essentially dihydrofolate reductase inhibition. But this is what essentially this is essentially what's led to these discovery of these mechanisms. And I'm quoting here from the paper in rheumatology that says it's important because it's providing considerable insights into the inflammatory properties of rheumatic diseases at the cellular and molecular level. But this knowledge has also helped explain some of the toxic effects of methotrexate and known side effects. So if we get into side effects and toxicity, which is why it's important to look and monitor this, there are mild side effects that are gastrointestinal, like nausea, upset stomach, mouth and skin issues, and then neurologically mild, like drowsiness, fatigue, headaches, or a foggy feeling. And on the severe side, it can lead to infection, bone marrow suppression, liver problems, lung respiratory problems, kidney, severe allergic reactions, and severe neurological symptoms like confusion, seizure, loss of vision, and weakness on one side of the body. So that's why it really is important to monitor methotrexate therapeutically, and it does have a narrow therapeutic window. And I'm sharing here the relevant clinical cutoffs and the decision points for physicians that are generally reported that you must be aware of for potential toxicity. We report something similar in our interpretation chart. Basically, if your methotrexate's over 10 micromole per liter after 24 hours after a dose, high risk for toxicity. After 48 hours, it should be less than or equal to 1. After 72 hours, it should be less than or equal to 0.1. So if it's greater than those cutoffs, it can be a high risk for toxicity. And it's also important to monitor for treatment to make sure even that higher doses, that as the patients are being treated for toxicity, the values are dropping. So this is to imply that it's not only that it matters if it's at just above 10, even if it's in the hundreds or the thousands, it matters to monitor and make sure treatment is effective. And it's also very important to recognize how we treat methotrexate. And I'll mention this in more detail why, but there are two major ways that it's treated. Commonly is what's known as leucovorin rescue therapy. So in this case, it's just to replenish concentrations of folic acid. And in more advanced and severe cases, or in patients who basically can have a reduced kidney function, there could also be administration of glucarpidase therapy, which essentially breaks down methotrexate itself into less harmful metabolites. So we'll come back to this point. So just remember, there are two therapies, leucovorin and glucarpidase, and we'll talk about why that matters. But now let's briefly go into how we measure methotrexate today. And there are two main ways we can measure them. One is immunoassays, like the ARK diagnostics kit we're using. It's typically a homogeneous enzyme immunoassay, and it's essentially compatible with major automated analyzers. They're typically FDA approved or cleared, and they can provide rapid results within an hour, and but can be affected by important interferences, which we'll get to in a bit. Whereas the other option is also an LC-MS based test that is obviously an LDT. They're not FDA approved. They're laboratory developed tests. They're not available in many hospital labs, but they're available through reference labs. But the problem being then if you're sending it out to that reference lab, you're going to have a slow turnaround time, and it may take 3 to 5 days to get that result, which may not be very useful in the immediate management of most patients. So it can be an issue, but they're very helpful. And when you're unsure of a result or there's an interference, LC-MS is the assay of choice because it basically helps eliminate that. They're highly specific and helps eliminate those interferences. But those are essentially the two main ways you can test methotrexate today. And focusing on immunoassay at hand, the ARK methotrexate from the product insert of the manufacturer, the measurement range is 0.03 to 1.3. At this point, I hope now you're thinking back to what we spoke as the clinically important ranges. We said even up to 10, obviously, and greater than 10 is important. So that's why you have to look at like doing additional dilutions and to get the results within that range. Precision is less than 3% at all tested concentrations, which is great. It's an excellent precision profile. Interferences from other methotrexate metabolites that are important to recognize, there's really minimal effects from 7-hydroxymethotrexate, which is a primary metabolite. But there is significant cross-reactivity with DAMPA. Now, DAMPA is normally not circulating in high concentrations except following glucarpidase rescue therapy. So this is important to recognize that in most patients, or those who have had glucoverin rescue therapy started, this is not an issue. But those who have had glucarpidase rescue therapy started, DAMPA rises, and that can have up to 50% cross-reactivity with the assay. So you could end up with falsely high methotrexate result after therapy started that can be confusing to clinicians because they're treating it and they're monitoring levels, and all of a sudden they don't see it go down, or it doesn't go down as much. That could be because DAMPA is present. So it's important to this interference. Now, when we validated our methotrexate assay here, we compared to a different immunoassay from a different manufacturer. And just I put this up here to say that they generally compare well within 10-15%. There's a slight bias on the upper end that's about 10-15% with a standard manufacturer assay. That was our alternate option based on the instruments we had here. So ARK compares very well with other immunoassays. And more importantly, though, versus LCMS, which is really the gold standard, there's an excellent correlation there. And you see that here, basically with a slope that's very close to the linear line with some variation that's expected along across the range. More importantly, and this is kind of key for us, because when we were considering, okay, which assay do we use? Do we go with ARK's kit, or do we go with the other IVD manufacturers' kit that we could have on the line? It's important to recognize that, when we looked at our data and the distribution of results, over the last year, based on 1,555 methotrexate results we've released, a third of them, 33.5%, are above the analytical measurement range of the assay of the other IVD manufacturer. Now, again, ARK was 1.3, so it's not that different. It's the same conclusion. I just highlight that here. But importantly, other limits by other manufacturers, their clinical reportable range, which is the maximum you could release using their kit, still leaves about 16% of your patient results that you could not resolve using those kits, and even after dilution. And I will add, some of those dilutions, even be within that, require manual dilution by a technologist. So that means that you will have to basically physically pull that sample off the line and do additional dilutions to get those results for about 20-25% of those samples. So that's not a trivial number that you'd have to manually treat. Whereas with what we've done and the dilutions we've implemented, when we do a 250x dilution on the ARK kit, we're able to release all results and increase our reportable range up to 12,000, which we've never had a sample, at least encountered in the last year, that was above that. So we were able to fully automate that process. And how we did this is essentially we built two separate applications for ARK on our automated analyzer, where one runs the standard AMR up to 1.3, and then it runs an additional 10-fold dilution if the value was greater than 1.3. So we were able to release with that first application results up to essentially 11.3 when you do 10 times that. But then the second application, if it's still greater than that, would automatically, or greater than 13, sorry, would automatically run the sample on a second application that already has the dilutions built in at 100x when it's running at the first time. And then it triggers even at 250x if it's still greater than 130 after that first run. So we're basically able to completely automate the process and not have any manual technologist intervention to release those results. So that's something that we could essentially be able to do this but with ARK, but we can't with our other IVD manufacturer because they restrict our ability to change any dilutions and basically adjust the applications to do this. So that was dissatisfying for us because we did not want to be manually diluting that many percent of our samples. That would be very high. So how do we handle samples from patients on glucarpidase? That's another big issue that we've mentioned is an issue with all immunoassays, unfortunately. So in this case, the recommendation is to send it to a reference lab that does testing by LC-MS/MS. And I'm just highlighting this as an example from one major manufacturer, so you're aware. When you go look this up, they even have the test named as methotrexate post-glucarpidase. So that's directing people to
Thank you, Joe, for a very insightful presentation. I'm sure there'll be questions. We'll come back to you with that, but that was really good. Thank you. Our next speaker today is Dr. Tom Hoots. He has over 30 years experience in research and development, innovation and program leadership across diagnostic and life sciences industries. He earned his bachelor's degree from Stanford University and his PhD from the University of London. Tom began his career with Siva Dade Behring and subsequently held a variety of different scientific and leadership positions at industry-leading organizations, including GE Healthcare, Amersham, Cyphergen, Affymetrix, Beckman Coulter, where he was the VP of Chemistry Development and Director of New Technology and Innovation. He's been with ARK Diagnostics in a senior role for about 9 years now. He has led program management and special projects here at ARK Diagnostics and is helping drive strategic initiatives, cross-functional execution, and advancement of new technologies. When Tom is not solving the complex scientific challenges, Tom is pursuing another passion in chemistry. He's a certified beer judge and is a very accomplished home brewer. So next time, Tom, you do have to provide some samples to us folks as well to prove that. He enjoys applying the same scientific principles. It's all in the beer brewing process. Outside of his lab and brewery, he also raises alpacas on his properties. So with that, I'll hand it over to Tom to do his presentation.
All right. Pankaj asked me to present on how we achieve the performance that we have. And I'll give you a little background on ARK and the method development. So let's to the next slide. The discontinuation of the Abbott TDX platform in the 90s created a market opportunity for ARK. And we launched our first generation of the ARK methotrexate assay in 2011. And at that time, we were the performance leader. But we set out to increase our performance leadership because we thought it was important clinically and for the business. So we began exploring monoclonal antibody conversion in 2014, and the goal was to achieve simpler manufacturing and much better performance. We had clear goals focused on the clinical utility of the test. There are really, the medical decisions range from around 0.05 to 0.1 micromolar, and throughout the range up to greater than 1,000 micromolar. And this means that achieving analytical measurement performance that's a little bit wider would help customers with dilutions and help customers get results quickly. We also wanted to make sure that we improved precision over the full analytical measurement interval. We really want, if you have excellent precision, it's easier to achieve all of the other performance goals. So we focused on that. We wanted linearity improvements and specificity improvements. And with all of this, we were also had some internal goals to make the assay equivalent or better stability, simple, consistent manufacturability, and eliminate REACH-listed preservatives. So our approach was to analyze the research and development data frequently for precision profiles over the entire analytical measurement interval. Precision profiles have been around since the 1970s, particularly for immunoassay, and they analyze precision as percent CV versus concentration. And they're an excellent way in a snapshot to see quantitatively how good the assay is going to be. So we used that as a tool. We analyzed frequently for linearity. And if we encountered any compromise on specificity, we redirected the project to get rid of it. So we focused on all of the clinical aspects. We designed the assay for comparison with LC-MS because that was necessary if we were going to use an expanded range. We made changes. We switched from rabbit polyclonal pools to a monoclonal antibody, but it took patience and persistence. It took 6 rounds of cloning. We worked with 2 different species. We screened hundreds of clones. We thought we were there several times and then had to go back and rework our approach on achieving the monoclonal antibody. And the specificity for methotrexate is challenging. I'll have a few slides later on that, but Dr. El-Khoury has already raised some of the key important messages there. We also made significant changes to the hapten enzyme conjugate. Many of them are proprietary, but we switched to a recombinant enzyme, which was easier to work with in manufacturing and had somewhat better performance. And we simplified the chemical reaction conditions. We also, those improvements allowed us to achieve improved calibration curve size in terms of millia, the span of millia per minute, which led to better analytical precision. And we have had improved stability as a result of what we're doing here. This is an example of precision profiles. Typically, they're U-shaped. We did a side-by-side comparison of the ARK assay with a major competitor. And really, this is a 20-day precision study, 1 run per day, 20 days, duplicates per run. We can see that the ARK assay is better than about 5 or 6% from 0.05 micromoles per liter to 1.3 micromoles per liter. So we generally, adjusting the antibody concentration will shift where the curve tilts upwards in the U shape. The competitor's assay is very good at high concentrations, but it is markedly inferior at low concentrations. So being able to have excellent performance over the entire measurement interval is key because the decision to dilute the specimen is important. There's a medical decision points at around 1 micromolar, and 0.05 to 0.1 micromolar is the region where it's safe to discharge the patient. Specificity is key, and why it's challenging is that the methotrexate metabolizes to 7-hydroxymethotrexate, and that's only a change here. And the concentrations can be very high for folinic acid or leucovorin, and because when there's delayed clearance, they increase the level of the rescue agent leucovorin. So we have no detectable interference from 3,000 micromoles per liter of leucovorin. It has more changes over here, and the methyl group is not here, but leucovorin, as mentioned before, bypasses it gets into the cells more easily, and it bypasses the blockage in the DNA synthesis from the lack of dihydrofolate or tetrahydrofolate. The other challenge, and this is just a little bit more information about glucarpidase, but glucarpidase cleaves off the glutamate residue, and it generates DAMPA, which really differs only from the loss of the glutamate. And so it quickly reduces methotrexate, but generates DAMPA. The DAMPA is shown here. It was tested by HPLC, and it rapidly peaks and gets close to the concentration of the original methotrexate, and then curves. And these horizontal lines here are the limit of detection for a limit of quantitation for the HPLC. The methotrexate concentration by HPLC comes down quickly, but then it's released back out from the cells and is sustained over time. Immunoassays, and this publication was before the methotrexate 2 assay, but all immunoassays suffer from cross-reactivity with DAMPA, which causes them to be high for many hours. So 4 days before it gets even close to the actual methotrexate level. So this period, it makes it really unsafe to be measuring with an immunoassay. ARK is better than other immunoassays, but it's still not good enough to be recommended for use after glucarpidase. As far as linearity and correlation with LC-MS, the ARK method met all of our goals, excellent performance all the way from the bottom to the top end of the analytical measurement interval, and linearity within 10% over the entire assay range. So the ARK assay delivers better performance with a broader assay range. Total precision at the control levels of better than 4%, minimized cross-reactivity from 7-hydroxymethotrexate, and it's consistent quality because the rabbit monoclonal antibodies can be generated in whatever volume is required, and the recombinant enzyme conjugate helps us make very consistent conjugates. So the performance has been published last year at the IATDMCT, International Association for therapeutic drug monitoring and clinical toxicology. And you're able to see the poster on the ARK website. Here's a QR code that makes it easy, but it's got the summary of the performance. And with that, I will stop the presentation and hand it back to Pankaj.
Thank you, Tom. That was really good. I think I brought in a lot of the efforts and the scientific innovation behind the assay. So thank you for that. Appreciate the presentation. If there's any questions, I know we are early right now, but I'm sure you have some questions for our eminent speakers. Maybe I'll get started if that's okay, Joe. And I was thinking about your presentation while you're presenting. Since you've implemented the assay, Joe, have you observed any changes in the clinical decision making or the patient management for you in your hospital setting?
So full transparency for us. So we are in the process of transitioning from the ARK 1 to ARK 2. So this is the validation data that I've presented. So I cannot yet comment, obviously, on the clinical impact of doing that. But we will be, hopefully, in the next month, transitioning over to the ARK 2 assay. So I cannot yet comment on that piece, if you like.
No problem. Another thing I noticed in your slide where you had the preparation of the groups, Joe, there was about 33%, I think it was 1.2 and above, and then there was a certain group. I think just for our, people who have logged in, is this something from your experience? Is it because of the nature of your hospital, because of the patient population that you have that, or would that be similar to other hospitals? Obviously, there's some focus to cancer, and there's some more general, and they have certain. But what are your comments on that in terms of the data points that you shared? Is it more pure Yale, or can also be thought about as having some kind of a universal generality to that data point?
Yeah, no, excellent question. So, as mentioned, you have cancer, for example, would be usually higher doses are used than treating psoriasis, which we talked about, low doses methotrexate, right? So for us, we are affiliated with the Smilo Cancer Center. So we have a major academic hospital affiliated with a major cancer center. So naturally, we see a lot of those patients, but I would say it would not be dissimilar to other academic centers or simply cancer centers. So I would expect this to generalize to other major academic centers, but not necessarily to hospitals that may not be affiliated with cancer centers.
Got it. I see a question from a gentleman from St. Jude, and you probably know him as well, Dr. Molinelli, and he said, for Joe, what instrument was used for the ARK2 test? Have you observed different performance between the instrument vendors?
Thank you. Yes. Hi, Alejandro. So we use Roche Diagnostics as our major IVD manufacturer for the automation line that we use. So the comparison we did was between ARK and Roche. And to be clear, we did not bring in Roche and put it on our analyzer to do the testing. We actually sent it out to our reference lab. And I'll highlight because I showed the slide. So we sent our samples to Mayo, which does the testing by both Roche and LCMS. LCMS is what they do when you order the post-glucarpidase methotrexate, as I was showing on that slide. So we basically did ARK versus Roche, and that's the comparison I showed you in that original data set. And again, they compared very well, except there was a 10% bias on the upper end, which could be calibration. I don't necessarily think that that's a problem with one assay versus the other. It just happened to be that difference on that day, because I don't think the methotrexate is not one that is standardized across manufacturers. So I'm not as surprised or concerned, but there was I'd say a 10% bias on the upper end in that comparison. But that's all that we observed as a major difference. We did not perform precision studies or any additional other studies on the Roche assay, but the data that Tom showed us also kind of compares that precision profile. So you can kind of get a sense where they compare, at least in terms of precision as well.
Alejandro, hope that answers your question. Otherwise, you can hit him up at ADLM and ask him again. But also, what percentage of patients, Joe, do get they get the glucocorticoid treatment? What percentage of patients get that? Is there a number you think, or?
I do not have that number, unfortunately. So, but it is rarer, and for individuals whose methotrexate level is not going down with glucocorticoid therapy, and typically it's reserved for patients who have reduced clearance. So patients who may have acute kidney injury or chronic kidney disease, there they may want to intervene and also give that to break down methotrexate more rapidly. So I don't know. I don't have a number. I have not looked at what % actually receive that treatment, but that is an excellent question.
One more, and Tom, you can chime in as well. You have some international partners in different parts of the world. It's pretty late there, but some major cancer hospitals, I can see their names. One of the questions I was asked before was, and this is more maybe clinical, but also from a lab perspective, is what is the frequency of testing for methotrexate? Is this driven by the clinician for pediatric population for children, or is there certain guidelines that the lab follows, or is it more clinician driven? Is it the same for kids or children, or same for adults as well?
So my understanding is that the protocols for when to test, what time points after the administration of the dose, that is generally specified by the clinical protocol. So the actual treating physicians, treating oncologists are the ones who decide what the testing pattern is. One of the things that I don't know is whether every specimen arrives with the lab with the information of how long it is since the dose was administered for the high dose methotrexate, which I think is important for the lab to really be able to comment on the interpretation and whether there may be evidence of delayed clearance.
That's great.
So
Thank you, Tom, for that.
a simpler answer though is that for each treatment cycle, you'll typically get at least 3 measurements, right? One at, say, 48, one at 72. Sometimes there's also a 24. But that's, I guess I gave the complex answer first. But anyway, Dr. El-Khoury, do you have any additional comments on that?
No. I think you captured it nicely.
There's a question from gentleman from Sanford. I'm going to read it out. We get requests from PEDS hemonic providers to report MTX values of 1,000. Couple of questions on the clinical reportable range. Number one, what is the maximum dilution, auto/manual, that may be practical to verify for MTX2? Second question, are there any recommendations on strategy for verifying dilutions? There are not many assayed material. There's a question mark to that. There's a third question, and I can repeat that later if you want, Joe. Are there any PK modeling strategies, or PK modeling studies confirming the clinical utility of MTX above 1,000?
All excellent questions. So I'll start with the clinical reportable range for the MTX2. It's important to state that in the product insert, and Tom can correct me, Pankaj, feel free, as I've read it, there is no, the AMR is listed up to 1.3, and then it says you may dilute, but there is no limit on the dilution and no specification on what you do for the dilution. It's up to every laboratory to validate the dilution they choose to use, which is different from other major IVD manufacturers who lock down even the dilutions and have limited the reportable range. So that's a key comment, and why we chose to validate the 10x on the same analyzer and then the 100x and 250x on the second application. So we basically validated up to 250x using that would allow us to release results up to 12,000. The second question is how to validate those dilutions. There's a number of strategies you could use, which could include getting methotrexate and doing dilutions and then testing those and proving that that works, or validating the dilutions themselves by getting a patient sample with like a high value within the AMR that you then dilute to 50x, and hopefully you get reliable result on the lower end. And if that passes, you've essentially validated the dilution itself. All of these are strategies. Some of it is well described in CLSI guidelines for validating an extended range. So it's basically what these are like two common strategies of how you could do it. It depends if you have access to highly concentrated stock solution of methotrexate to prepare the solution. And if you don't, you could do the second approach, which is diluting within the range. As long as you get reliable results on the high sample and the low samples within the AMR, you essentially validate the dilution. And then the last question, and I'm trying to recall, are there any pharmacokinetic studies demonstrating the utility of results greater than 1,000? Not that I know of. I know our clinicians care that when they're treating a patient who has high methotrexate, they want to be able to monitor and make sure it's going down, or if they need to have any more severe interventions, like giving any of these more advanced therapies to basically lower it even further. So that's essentially the clinical utility that I'm aware of, but I don't know of any pharmacokinetic studies. But hopefully that covers it, Pankaj.
Yeah, I think so. It's very comprehensive.
I'm seeing comments in the chat as well.
Chat
if separate from the Q&A. Yeah.
Right. Yes, I just logged in there. So there's a question from Melinda Renat Gonzalez. I'm not sure what organization, but I'll read out the question. We're currently performing methotrexate testing on the vitro system using the original methotrexate reagent. I was informed that methotrexate 2 has not yet been validated for use in vitros, therefore not currently supported by QuidelOrtho. Although this may be outside the primary topic, I would appreciate some clarification. That's probably for us then. Is Jackie
I
or
can
Tom,
answer
can you confirm that one?
Yeah.
Yeah, please. Thank you.
So we have completed the validation testing, and so from our perspective, it's validated. I don't know where we are as far as QuidelOrtho commercializing it as far as their distribution system.
Hope that helps, Melinda. Please feel free to reach out to us and we'll be in contact about that. There's another question from Jane Shale, and this is for Joe. If MTX was overdosed accidentally and not treated for weeks, what is the extent of the damage on the human body?
I'm not a clinician or a toxicologist who works with these patients, but generally from the literature review I've done is it can be fatal even in extreme cases. So that's why it's important. And I would doubt that it would progress to that stage being untreated, because there's severe symptoms that I mentioned that would be clinically obvious that would generally bring a patient back to the hospital, I would hope. But yeah, the consequences are severe. We've talked about kidney issues, liver problems, respiratory problems, bone marrow issues. So including severe neurological symptoms, all of it would be clinically obvious symptoms that would hopefully bring the patient back to the hospital, they would get treated for it. So generally, even in the absence of having a methotrexate level, they would treat the clinical symptoms. So the goal would be then, if glucocorticoid therapy hasn't started, they could get the immunoassay result and confirm. If that has been started and these are developing, they may send to LCMS, but they would treat the patient for those symptoms while confirming. At that point, the LCMS results is just confirming what they observed, because it doesn't come back within 3 to 5 days if they're sending it out, unfortunately. And I'm seeing,
Thank you for that, Joe.
Yeah, is there a new Q&A or? Yeah, that was the previous one. Okay.
Yeah, we have Joe's pretty famous, so you guys know how to reach him. But if you're not able to, we can provide his contact information. I think you had it in your last slide probably, but he'll also be at ADLM. But if there are any other questions, please feel free to reach out to us. Dr. Hoots and Dr. El Khoury will be happy to respond. I'm pretty sure about that. in terms of ADLM, our diagnostics will be there. We'll be at booth 2526. Please come and see us there. We'll love to share more information on the MTX2 or any applications or questions related to that for your machine and any other scientific inputs that you may want from us. But I guess I don't see any other questions. If there aren't any more, then we can conclude our webinar. But before that, thank you, Joe, very much for making time. I think this was extremely informative. I think everybody who attended will benefit from it. And I can tell you there was a lot of people, scientists from Europe, who were mildly disappointed that we scheduled it at this time because there's probably the World Cup time there watching some game or later in the evening, but they have all committed to watching a webinar and a recording of that. So you might get some questions from our European scientist colleagues as well.
It's very hard to compete with Holland in Norway, who are playing right now.
That is true. That's absolutely true.
Sorry,
I think we're maybe one more. Sorry, I think we have 5 more minutes. Let me. Okay. We are currently running the original MTX assay on the Alinity, while the packaging source is AMR is 1.3. The build parameters for the upper linearity set to 1.25. The defined MTX had higher results in validating onboard manual dilutions. Did you see that, Joe? Or
yes. So I don't know why on the Alinity is set to 1.25. I know on our current Roche analyzer using the open channel setting up ARK, we did set it up to 1.3, but that's not a major difference. I think the dilution question, which would be more important to expand that range, is all of the dilutions we perform are automated. So that's basically what we did by doing an automated 10x dilution if the results are greater than 1.3 built into the application. And then we created a second application where we changed the volume in the application of the sample to the reagent so that it's automatically running at 100x the first time. If it's greater than the 13 based on the initial run, it reflexes to the second application where it runs on 100-fold dilution essentially, and then if it's still higher, it runs it at 250x dilutions. So that's how we did it, and we were able to validate, as I mentioned previously, using the approach recommended by CLSI by validating the dilution itself. So we ran it essentially in triplicate several concentrations on those high end to show that it reproduces at 250x dilution and 100x without a problem. I hope that answers it, but I don't know why Alinity would be 1.25.
Kirsten, I'd be happy to answer your question in more detail if you email me or phone me, but my email is tom@ark-tdm.com.
Thank you, Tom. Well, I think on that note, we'll say thanks to everybody for joining, and we hope to do subsequent webinars with industry leaders and lab medicine leaders in the coming months. But thank you all for making time today to listen to Joe and Tom, and big thanks to both of you gentlemen for the webinar presentations. Have a good day, everyone. Thank you.